All right,
Yep.
thank you, Suzanne and Steve, and the Immcantation team for inviting us to present some of the published work from our lab. I'm Sureshchandra, a postdoctoral fellow in Lisa Wager's lab in UC Irvine. And today I'm going to be talking about the utility of the tonsil immune organoids to look at B-cell responses to influenza antigen formats. So influenza affects 3 to 5 million people and resulting in hundreds of thousands of deaths every year. But the vaccines against influenza is the primary way of preventing infection. The effectiveness of the vaccine, however, is quite variable, as you can see in the graph on the right. The effectiveness ranges from roughly 20 to 60, depending on the year. So to some extent, it reduces morbidity and mortality, but the vaccine is not fully effective. And currently, what we know from the influenza field is the presence of neutralizing antibodies as the sole correlate of protection against the vaccination. While profiling antibodies in blood is great, the actual B-cell responses and T-cell responses against vaccines evolve in these structures, microanatomical structures called germinal centers. So these germinal centers are restricted to tissue sites, so primarily in lymph nodes, tonsils, and spleens. And there are unique players in the germinal center. As you can see on the right, this cartoon, looking at different immune cells that are involved in the initiation as well as evolution of adaptive immune response. So T cells interact with DCs, go into the germinal center, interact with the germinal center B cells, specifically light zone B cells, and they go into the dark zone within the germinal center where they mutate and proliferate. And there is evolution or production of plasmablasts that produce high affinity antibodies. So a lot of these cells are quite restricted to the tissue sites. So to identify correlates of protection from sampling just blood alone is quite challenging because these cells very rarely circulate in our body. While some labs have been very successful in sort of looking at germinal center response in humans, like Ali El-Beri's lab that looks at responses post-vaccination in lymph node aspirates, and Donna Farber's lab that looks at T-cell responses in postmortem tissues, it's very challenging to do high-throughput experiments in humans, particularly when you are interested in looking at germinal center responses. So my PI, Lisa, she innovated this technique of tonsil immune organoids, where we isolate lymphocytes from discarded tonsil tissue, and this could be tonsil or spleen or lymph nodes. So we isolate lymphocytes and cryopreserve for long-term storage. And we were able to revive these cells in batches and perform generate organoids that last for 2 to 3 weeks. So you're able to look at long-term cultures, T and B cell responses over the course of 2 to 3 weeks. And we use classical immunological techniques like flow cytometry and ELISA, and have been recently using single-cell RNA sequencing paired with TCR and BCR analysis to look at antigen-specific T and B cell responses. So in the original study, as proof of concept, demonstrated that in the presence of the vaccine, so here I'm showing LAIV, which is a live-attenuated influenza vaccine. In the presence of the vaccine, you're seeing an expansion of plasmablasts. And then on the right, you're seeing the presence of antigen-specific antibodies. Like in humans, the responses here are highly variable. There are high responders and low responders, but you also have a lot of donors that have been either recently infected or vaccinated, resulting in high background in the unstimulated cultures. So we're able to look at antigen-specific responses for roughly 2 to 3 weeks, here 21 days. But we're also able to track antigen-specific T cells. So here we're looking at flu M1 specific CD8 T cells that expand post-LAIV in cultures in one donor. So the advantages of human immune organoids are somewhat obvious. You can look at temporal GC responses, which are quite challenging in humans in vivo. It allows us to do mechanistic experiments. You're able to sort of remove or enrich specific cell populations on day 0, generate organoids with or without those cells, and look at their role and how they contribute to an evolving germinal center. And in my opinion, the most important advantage is the throughput of the system that allows you to look at multiple formats of the vaccine and virus or adjuvants in the same donor, which is not possible in humans. So since the first publication, our lab has sort of moved on to looking at other factors that regulate vaccine effectiveness. So one arm of the lab looks at host features. So looking at age, sex, immune history, and how all of those can influence vaccine responses. But a more challenging aspect is to understand how the vaccine format itself is regulating vaccine responses. So if the vaccine is intranasal versus intramuscular, if it's a subunit vaccine versus an attenuated vaccine, how are those features of the vaccine itself regulating adaptive immune responses? So currently for flu, there's two vaccines. There's the inactivated influenza vaccine, which I'm going to be calling IIV throughout the talk. This is a subunit vaccine that has been approved, that is approved for adults in the US, which basically uses purified viral components, the hemagglutinin part of the virus, and is given it's an intramuscular vaccine. Whereas you have LAIV, the live-attenuated version of the vaccine, which is prescribed for really young children. It's a cold-adapted version of the virus that has all the components of the virus, but is attenuated and does not replicate, and it's given intranasally. So there are two versions of the vaccine, which I'm going to be talking about today. So the study was pioneered by a previous postdoc in the lab, Jenna Kastenschmidt. So the question we're asking is, how does the antigen format, the two versions of the flu vaccine format, influence adaptive immune responses? So we started with a cohort of adults. So we started with adults because we know they're predominantly exposed to either the vaccine or the virus, or both. And we generated organoids from the tonsils from these donors and stimulated them with either IIV or LAIV or the wild-type virus itself. So we used the 2009 A California H1N1 wild-type virus to stimulate and generate organoids and profile their T and B cell responses on days 4 through 14 using single-cell RNA sequencing and flow cytometry for the cellular aspect of it. But we also used protein microarray, ELISA, and neutralization assays to look at the antibody side of it. We started by looking at antigen-specific B cells. So this is just a flow plot to look at different subsets of B cells using classic markers 27 and 38 in the germinal center. But we're also able to look at antigen-specific B cells. So using a bioattenuated version of the hemagglutinin, we're able to look at HA-specific B cells within the population, within the organoids in every single donor. And we're able to look at the phenotype of those cells as well. So in the graph below, you're looking at HA-specific B cells that is within the plasmablast population. As you can see, LAIV induces much higher proportions of antigen-specific B cells compared to IIV. And that correlates with the antigen-specific antibodies that have been measured using ELISA in the conditioned media. So LAIV is the magnitude of antibody response in the same donors is much higher in LAIV compared to IIV. And that to some extent correlates with the neutralization, the graph on the right. Here, AUC is the inverse of neutralization. So LAIV is much higher, induces much higher neutralizing antibodies compared to IIV. So in general, LAIV results in a higher expansion of antigen-specific B cells, antibodies, as well as the quality of antibody. And this is not restricted to just the HA-specific antibodies. It also goes to neuraminidase antibodies. So here you're looking at protein microarrays. So these are antibodies against hundreds of flu proteins using a microarray. And what you're seeing here is the aggregate of those, the signal of those antibodies. So you're looking at HA from different strains of the virus or even influenza B. In general, LAIV induces broadly cross-reactive antibodies compared to IIV. And then we use single-cell RNA sequencing to look at both the cellular aspect of B cell response in higher resolution, but also be able to look at the BCR in parallel. So here you're looking at the UMAP of roughly 100,000 cells, B cells that have been sorted out from the 4 stimulation conditions from 4 donors and 4 different time points. There's T-cell aspect of it as well, which I'm not going to be talking about today. But in general, you're looking at populations of B cells that you would expect to see in germinal center, memory cells, dark zone, light zone, and plasmablasts. But we are so able to see 2 transcriptionally distinct populations of plasmablasts, PB1 and PB2. Now, PB1 was a population that was transcriptionally looked like it was short-lived, and it expanded in IIV cultures pretty early on, whereas PB2, they look transcriptionally like long-lived plasmablasts. And these are expanding in LAIV much later on. So you're seeing an early expansion of plasmablasts, or at least antigen-specific plasmablasts in here. The graph in the middle is actually looking at antigen-specific B cells. So on the left, the UMAP is all B cells from the organoids. And on the right, you have these colored dots that represent the antigen-specific B cell, because you're able to sort out the HA-positive cells, tag them with hashing antibodies, and sort of run them as samples in single-cell RNA-seq pipeline. But a unique aspect of the cellular response was these pre-memory cells that express high levels of FcRl4. So this population of cells were predominantly what IIV's antibody antigen-specific response was, and they are expanding in IIV pretty early on, but not seen in either the virus or LAIV. And the expansion of this FcRl4 is not an artifact of the organoid system. So we went back to the data from Ali Alberi's lab, where they have single-cell sequenced the B cells from lymph nodes post-IIV vaccination in healthy donors. And when you actually project the transcriptional profiles of that cluster of cells, we see a unique cluster of cells in the from B cells in vivo, and they express FcRl4, and the kinetics of those sort of replicate what we see in our cultures, that they are expanding roughly around day 18 in vivo, but we're seeing the peak happening in IIV. So suggesting that there is some sort of confirmation between what we see in organoids in terms of antigen-specific response and what's happening in vivo. And then we have the BCR data. So we are able to look at the somatic hypermutation, affinity maturation, and class switching in antigen-specific B cells. So here we use Immcantation tools, primarily Chango, to look at the gene segments, gene usage, as well as the isotypes. So on the left graph here, we're seeing different gene IGHV genes being used in IIV versus LAIV. So there's biases towards 169 in IIV versus 323 in LAIV. So we picked out these genes based on literature suggesting that there's dominance of those genes in broadly neutralizing antibodies. And on the right, you're looking at the isotypes. So this will help us in identifying the class switching in different subsets at different time points. So you have day 4, 7, and 14, but you also have 3 different subsets. You're looking at memory cells, GC cells, and plasmablasts, which sort of start appearing much later on in culture. So you have only day 7 and day 14. What we're looking at is different isotypes. As you can see early on day 4, the immune response is primarily, the B cell responses are IgM positive. But at day 7, you're seeing, at least in IIV, this dominance of IgG1, versus in LAIV, you're seeing a mixed response of both IgM and IgG1. So there is early class switching of antigen-specific B cells in IIV compared to a much later expansion of those cells, or switching of those cells in LAIV. And then we wanted to look at the clone sizes. So we used Alakazam to look at the diversity analysis. So here, again, by day 7, you're seeing large clones evolve in the antigen-specific pool of cells in IIV. Some of these are clone families of greater than 10 cells, but not so much in LAIV, where, again, the response is probably evolving. But LAIV catches up at day 14. So by day 14, you're seeing larger clones appear. But both the vaccines are performing better in terms of clonal expansion compared to the wild-type virus. And the graph in the bottom, you're looking at somatic hypermutation, sort of giving us some indication of what of affinity maturation. So early on by day 4, we don't see a lot of somatic hypermutation. So the response is still evolving. By day 7, IIV, the mutation rates in IIV is much higher than LAIV or the virus in both the memory cells as well as the plasmablasts. And by day 14, LAIV sort of catches up, but they're both higher than what we see in the wild-type virus. So collectively, what we've seen both at the cellular level and the BCR level and the antibody level is that IIV has a more directed and restricted response that might be more dependent on memory. So it's calling in a pool of precursor memory cells. They are clonally expanding very early on and class switching very early on, versus LAIV has a much more diversified response that relies on both naive and memory compartment. And to test that, we sort of did a depletion experiment where we started off with the wild-type conditions where you have the native proportions of memory B cells, but you also have cultures where we've depleted the memory B cells. So in yellow, you're seeing cultures where the memory B cells are absent. And then we generate these 2 conditions, sort of mimicking what you would ideally do in a mouse setting, and then stimulate them with the vaccine antigens and culture them for 7 days. As you can see in both in terms of antigen-specific B cells and antigen-specific antibodies on the right, IIV sort of attenuates depletion of memory B cells sort of attenuates the antigen-specific response in IIV, but not so much in LAIV, where you still see some sort of evolving responses. So suggesting that memory B cells are absolutely essential for generation of B cells, antigen-specific B cell response in IIV, but there might be in LAIV. This sort of holds true for CD4s as well. I'm not showing a lot of T cell data here, but it's in the paper. But the overall, the study sort of summarizes that in IIV, which is subunit vaccine, you're seeing poor type 1 interferon response, very limited plasma-blast differentiation, but you're seeing early restricted BCR repertoire. You're seeing expansion of specific clones and more likely coming from the FcRL4 pre-memory cells, which leads to limited strain-specific antibodies and poor neutralization overall, versus LAIV, where you're seeing a strong type 1 interferon response, expansion of plasmablasts, more diversified antibody response seen both at the BCR and antibodies, and leading to greater neutralization and a more diversified response. So I hope I've convinced you that the tonsil organoid system is a great way of actually looking at immune responses in humans, particularly if you're interested in looking at T cell and B cell responses and the interplay between T and B cells, particularly the T follicular helper cells and germinal centers, which are very hard to track in blood. And the study sort of highlights that the two versions of the vaccine have very distinct cellular contributions, particularly in the B cell side, that leads to different neutralization and breadth. And with that, I'd like to thank the lab, particularly Jenna Kastenfish, who pioneered the study, and our collaborators, funding sources, and the tonsillectomy patients who provided us with the tissue. And I'd be happy to take any questions.
Thank you, Suhas. We have a few questions in the chat. I will just start with the first one. How did you differentiate the memory B cells from LLPCs? Is it the tissue localization or the surface markers?
The classification of memory B cells is primarily, so in the single cell RNA sequencing, we have surface protein staining as well. So you can actually look at CD27 and 38 and then classify them as either memory cells or germinal centers. So it's the surface marker, not the localization within. We don't have the spatial information yet.
You have a lot of more questions in the chat, Suhas. If you could take some time and go and answer them. And we are going to move to the next speaker. Thank you.